Marshall Nirenberg and Har Gobind Khorana's group cracked the genetic code. They figured out how the nucleotide language of mRNA is translated into amino acid language of proteins. Genetic data from Crick and others show that 3 nucleotides form a codon, an mRNA word that specifies one amino acid. This made sense because a codon made from only 1 or 2 nucleotides would not produce enough combinations, or words, to code for all 20 of the known amino acids. However, a 3-nucleotide codon produces 64 combinations. This would produce a redundant, or degenerate, code where several different codons specify the same amino acid. The parsimony principle, the simplest solution is often right, ruled out a 4-nucleotide codon. In 1961, Marshall Nirenberg and Heinrich Matthey started experiments to test the triplet codon hypothesis. They used a cell-free extract of E. coli with all the components needed to translate mRNA into proteins. The extract was treated with DNase to destroy any remaining E. coli DNA. So, there was no template from which messenger RNA could be made. Using a method perfected by Marianne Grunberg-Monago and Severo Ochoa, Nirenberg and Matthey made synthetic mRNA composed entirely of uracil. This poly-uRNA was added to the extract. Radio-labeled amino acids were also added. When the products produced in this cell-free system were examined, they found only polypeptides made entirely of amino acid phenylalanine. From this result, Nirenberg and Matthey concluded that a sequence of 3 uracils must code for phenylalanine. They had determined one of the 64 triplet codons. Nirenberg and Matthey then tried other polynucleotide chains. Poly-C made a proline chain. Poly-A made a lysine chain. Interestingly, no protein was made with the poly-G chain. Using RNA templates containing different nucleotides combinations, made by Har Gobind Khorana, about 50 triplet codons were assigned to their respective amino acids. On a genetic code table, finding a specific codon, look down the left-hand side for the first letter of the codon. For a second, look across the table. For last, look down the right-hand side. Some of the codons were hard to decipher because of their biochemical order. For example, a triplet with 2 GS and 1 C could be in the order CGG, or GCG, or GGC. To solve this problem, Phil Letter helped Marshall Nirenberg with rest of the genetic code. They took the advantage of the activation of tRNA. tRNA carries amino acids to the ribosomes for protein synthesis. In 1962, Robert Holley solved the structure of tRNA. Although tRNA is single-stranded, stretches of complementary nucleotides hydrogen bond to form short double-stranded regions, which bend the tRNA into a characteristic cloverleaf shape. Holley showed that all tRNAs have a similar cloverleaf structure. At a position on one of the leaves, a sequence of 3 nucleotides forms an anti-codon, which base pairs with a specific mRNA codon. Thus, there is a different tRNA molecule corresponding to each mRNA codon. Using their cell-free translation system, Zemechnik and his group showed that tRNA becomes activated when the amino acid binds to the tRNA stem. This step requires chemical energy in the form of ADP. Short RNA chains made of 3 or 6 nucleotides could be synthesized. When these were added to a cell-free extract, an activated tRNA will read this trinucleotide or hexanucleotide sequences. By separating the bound tRNA, the amino acid it carried could be identified. All that remained was to make specific trinucleotide and hexanucleotide chain to confirm the rest of the genetic code. In deciphering the genetic code, it was also found that translation starts with a specific codon AUG, the only unique codon, and there are stop codons that end translation. So, in addition to words, the genetic code also has punctuation points. So, to put this all together within the nucleus, the DNA code is transcribed into a complementary mRNA molecule. The mRNA enters the cytoplasm, where it associates with a ribosome. The mRNA code is then translated into a polypeptide chain. The codon AUG signals the start of translation. An activated tRNA ferries the first amino acid, methionine, to the ribosome. The tRNA anti-codon binds to the AUG codon on the mRNA. The whole complex shifts, and the next codon is read by another tRNA. The process is repeated until a stop codon is encountered. When the stop codon is reached, translation is finished. Stop codons do not have matching tRNAs. The ribosome disassembles to be reused for translating another mRNA, and one complete peptide chain is released. Marshall Nirenberg cracked the genetic code along with Har Gobind Khorana, who was one of the world's best organic chemists working in the field of nucleic acids, highly experienced and superbly equipped to do this. He synthesized the 64 triplets chemically, and he also synthesized repeating polymers with known doublets, repeating doublets or triplets as well, and towards the end he used them all to determine nucleotide sequences also. He used the ribosome binding assay that they determined all of the nucleotide sequences of RNA codons.